Just like the other three biological molecules, carbohydrates, lipids, and proteins, nucleic acids are polymers of smaller subunits called nucleotides. I did not make this Lego analogy, but I do love it because each Lego is a nucleotide. However, as you can see, these nucleotides have a slightly different identity than each other, as noted by the coloring of the Lego block. Each nucleotide, or each Lego building block, has three things in it: a sugar, a phosphate group, and something called a nitrogenous base. All three of these we delve into much more detail when we talk about making proteins. It is the nitrogenous base that gives these subunits their own identity. But what makes things a little bit more complicated with DNA is that DNA is really two polymers of nucleotides that are bound together by hydrogen bonds. And you can see that represented in the bottom picture here with the two stacks of Legos that are bonded together by dotted dots. They're bonded together by hydrogen bonds. The two main nucleic acids are DNA, or deoxyribonucleic acid, and RNA, which is ribonucleic acid. The difference between these two molecules is one oxygen atom, which is why DNA has the name of deoxy. Sneaky, sneaky. As mentioned before in another mini lecture, I have taught from books that consider ATP to be a nucleic acid. I don't necessarily disagree with that. ATP is technically made from a sugar, a bunch of phosphates, and a modified nitrogenous base. So I accept. DNA really has one purpose in your body. DNA is like an encyclopedia, right? I guess it's more like a recipe book that holds all of the recipes for all of the proteins that you might want to make. DNA also holds the instructions for making more cells. So DNA is like a storage molecule, basically. It stores information that dictates the sequence of nucleotides or Lego blocks. Conversely, RNA has more than a few functions, and we're discovering more of those functions all the time. Let's talk about DNA, and then we'll move on to RNA. Nucleotides are the building blocks or the repeating subunits of nucleic acids. And listed here are the 5 known nucleotides, which are categorized into two groups, the pyrimidines and the purines. As you can see, pyrimidines and purines have very different hydrocarbon ring structures. And it's this structure that allows pyrimidines to bond to a purine. A phosphodiester linkage is a chemical bond that forms the backbone of DNA, the repeating sugar, phosphate, sugar, phosphate, sugar, phosphate. It occurs when a phosphate group connects two sugar molecules in an adjacent nucleotide. And it does that through a bond called an ester bond. Specifically, the phosphate group links the 3', the hydroxyl group end of one sugar, to the 5' end of another sugar. This linkage creates a repeating sugar-phosphate backbone with the nitrogenous bases attached to the sugars and extending outward. Phosphodiester bonds are critical for the structural integrity of nucleic acids. In fact, it's these types of linkages that help form that twisted ladder structure of DNA. These bonds are really strong, but they can be broken during hydrolysis or enzymatic processes like DNA replication or transcription, which are two things that you want to happen. Those are not bad processes. Let's go back to our analogy of Legos for a moment. We have one molecule of DNA with two strands of nucleotides that are bonded together by hydrogen bonds, noted in this picture by these dotted lines. So we have one strand of nucleotides over here and another strand of nucleotides over here. Let's extend that analogy and think of DNA as a ladder. The rails of the ladder, or the place where you would put your hands on the ladder, are composed of the sugars and the phosphates that are in each of the individual nucleotides. And you can see the sugars here as the pentagons and the phosphates here as the P with the O's around it. The rungs of the ladder, or where you would put your feet, are made up of bonded nitrogenous bases of the nucleotides. Because of the specific nature of bonding of the strands, this structure does not stay as a ladder, but it starts to twist itself into a structure that is known as a double helix or a twisted ladder. The picture on the right shows you what's commonly called the ribbon model of DNA. But the picture on the left, it's actually what it really looks like. Note how the rails of the ladder are along the outside and with the alternating sugar phosphate backbone. And note how the rungs of the ladder are the nitrogenous base that's paired with the hydrogen bond. Why hydrogen bonds? They're weak. Why would you want to have weak bonds in something that basically holds the instructions for your life? Well, you need to access those instructions. And the hydrogen bonds make the DNA easy to break apart and easy to reform in order to duplicate it or to access the recipes to make proteins from it. RNA can be used as a storage molecule, but RNA is disposable, so it's certainly not for long-term storage. When it serves this purpose as a storage molecule, we call it mRNA because it's usually taking that information and taking it from one place to another as a messenger. mRNA is used to move information from the nucleus, where the DNA lives, to places further out in the cytoplasm of a cell. RNA also serves as a translation molecule, acting something like a language-to-language dictionary or a decoder ring. tRNA, or transfer RNA, has areas on it that can bind with mRNA, a sequence of nucleotides, and it can also bind with amino acids, which are the components of proteins. So it's almost as though the tRNA knows the language of proteins and amino acids, but it also knows the language of nucleic acids and nucleotides. And in that way, it can serve as a decoder ring. The last type of RNA that we cover in this class is rRNA, and we don't cover very much of it, which makes up a component of cells that are called ribosomes. Ribosome it's a lot like a protein factory. It takes individual amino acids, and with the help of tRNA and mRNA, it builds the specific protein whose recipe is encoded in your DNA.